Hello, everyone. Welcome to Nature and Science Podcast, where science does care what you believe. I'm your host, Don Murphy. We explore the science behind the latest technological developments, ask the deep questions about existence, and seek verifiable truth leading to a clear view of reality. On this episode, we speak with Dr. Jesse Goodman about the spread of the H5N1 virus, or the avian flu virus. We will discuss what a virus is, how viruses migrate from animal populations to humans, and assess the chances for a new pandemic. Dr. Goodman directs Georgetown's COMPASS program, which focuses on science-based policy and research to address unmet public health needs. Until February 2014, he served as the chief scientist of the U.S. Food and Drug Administration, a position he held in 2009, along with Deputy Commissioner for Science and Public Health. As FDA's chief scientist, he had broad responsibility for strategic leadership of cross-cutting scientific and public health efforts, including developing and implementing FDA's strategic plan for regulatory science and FDA's public health preparedness and response and medical countermeasures efforts. Dr. Goodman is a graduate of Harvard College. He received his MD from the Albert Einstein College of Medicine and did residency and fellowship training in medicine, infectious diseases, and oncology at the Hospital of the University of Pennsylvania and at the University of California in Los Angeles, where he was also chief medical resident. He has served on numerous advisory boards and committees for organizations including the CDC, the National Institutes of Health, the World Health Organization, and helped develop the research and development section of the Global Vaccine Action Plan. Dr. Goodman, welcome to the program. It's good to have you here.
Thanks very much. Great to be with you.
So on this episode, I'd like to begin kind of asking you about what inspired you to become a doctor in the first place, in particular, study viruses. That was an interest of mine from a young kid. I have to say that I followed Robert Gallo like he was a guru when I was a young kid and read all of his papers and was interested in type C viruses. Don't ask me why. It was just one of those things I was curious about as a youth. So I'm interested to know what got you so interested in all of this.
Well, I would say I came to viruses, kind of late, like after medical school and residency and all that. But if I go way back, more into the roots of things, I would say probably the biggest influence on me in going into science to begin with was my grandfather, who was a chemist and an inventor, really, and who actually started out as a biochemist. And when I was young and in high school and college, he was so enthusiastic about life science that, he really embedded this in me. And I was one of these kids who had, it wasn't a chemistry set, it was chemicals from his labs and, real Bunsen burner and all that. And it was sort of a miracle I didn't blow up my whole apartment building in New York, because I was kind of given free rein. But I sort of had an interesting combination of that, and then my parents, who were much more on the art side of things and involved in film and theater. So somehow I didn't go to college and I didn't leave high school intending to be a doctor, but somehow when I was in the university, these two interests combined, along with trying to make a difference in the world and in making me become really interested and passionate about medicine. And so no one was more surprised with that than myself or my parents, who said, "Are you sure that you want to do that?" So that's what got me going in that direction in medical school. And then, initially I actually was, I trained in internal medicine, and, you're a young physician, you are at the peak of your kind of book learning. And I felt like infectious disease was really interesting. The history was really interesting. The engagement with public health was really interesting. but I kind of felt like, well, everyone should know that. And initially, I started out actually working in oncology. So
Hmm.
this and ultimately on viruses. And I was really drawn to viruses by the person who was my lab mentor, who was named Jack Stevens, who was a superb herpes virologist.
Hmm.
And I was sort of interested in these viruses that could both cause infections and cause malignancies. And that's what drew me to it. And then ultimately, I just realized this whole world of infectious disease is so amazing. Uh, I'm going to take that oncology training, build another few years of fellowship on top of that, and be really overtrained. And ended up doing infectious diseases and first on viruses. And I worked on viruses for many years, and then actually ended up. I would say every 10 years I've kind of gotten stimulated or excited about something new, and it's kept me going. And then I had moved from LA to Minnesota and got involved in tick-borne diseases. But, those were what got me into science, virology to begin with, and that ultimately into public health and government and public service,
Hmm.
which sort of combined a lot of these interests and the realization that through public health and public service, I could take the science and have really broad impacts in improving the health, not just of my patients, but of the community and potentially the world.
Right. So let's start at the beginning. We talked about viruses, my own personal interest in virus, although I was interested in all sorts of bugs. I took water out of every pothole and pond I could find and put under my microscope, and was just fascinated by microscopy and little things that you couldn't see. So, what is a virus? I mean, I know it's everywhere in the lexicon. We've gone through COVID. People talk about it. And I know you're not surprised because you're in the business that still most people don't even know what a virus is. So let's tell them what a virus is.
Yeah, there's still scientific debate about what a virus is and where
Yes.
they came from. But I think probably the simplest way to think about it is a virus is probably regarded generally as the simplest form of something that you could consider alive. Although it can't reproduce on its own, it requires a cell to do that in. Some plant viruses, it might be a plant cell. There are viruses that actually infect bacteria. It might be a bacterial cell, and then there are viruses that infect humans, mammals, et cetera, and then it would be a mammalian cell. So the virus can't live, can't reproduce without that. It's a pretty simple thing. It has genetic information in the form of RNA, which everybody has heard of because of RNA vaccines, or DNA, which is our genetic information source in humans. And then it has usually a code around it, a protein, and some have another code or envelope around it. But so it's very simple, but it has to enter one of those kinds of cells, and then it uses its genetic information and the machinery of the cell to reproduce itself. And so it's not like a bacteria, which can just grow, or a mold, which can grow on a plate with the right nutrition. It requires a living thing in order to grow.
Right. So, and in this growing and in this taking over of the cellular machinery of either a bacteria or a plant or a human cell, when it's growing and it's producing more viral particles, that's when the disease happens. Is that right? When these cells are infected and then the viruses burst out of the cells and that causes maladies and various diseases.
Right. And I think that's the key thing that goes on, the hijacking the machinery of the cell to reproduce yourself at some cost to that cell. And sometimes the cost is like murder. The cell is killed. There's just so many, the machinery is taken over, the cell can't maintain itself. Maybe the virus puts holes in the membrane to leave the cell, et cetera, and the cell collapses. And certainly, if that's your cell that's lining your lungs, you could see why it could cause an effect like a pneumonia, or if that's a cell in your liver, it could cause a hepatitis. Um, but what's interesting is viruses also can do a lot of other things to cells um, and to human hosts and animal hosts. So, for example, the virus stimulates the immune response. We try to get rid of it. We try to make antibodies and we make chemicals called cytokines that try to stimulate our immune cells to fight off the virus. But sometimes that response of the host in defending itself can actually be harmful to the host as a whole. So, for example, in COVID, it's not
Right.
just the virus that causes the pneumonia and kills the person, it's also the host's over exuberant response in trying to get rid of the virus.
Right.
So it is quite complicated. And then some viruses, HIV is an example, um, the common herpes viruses and varicella zoster or shingles virus are examples of things where parts of the virus genetic information may persist, or the whole virus may persist in a cell. The cell may go on living, but in the case of herpes viruses, periodically the virus can act up and replicate and cause disease like shingles or like a herpes outbreak.
Right.
Or, in the case of HIV, it may be in the cells and, you know, integrated into the nucleic acid of the cell, but able to still replicate itself and with the cell still alive, not always kill the cell.
Well, I can tell our listeners that I'm a witness to the fact that these viruses can stay dormant. I just had a battle with shingles myself, and
Um,
it can be quite painful. But there are treatments, and that leads me to ask about antivirals because I know that most people know that viruses can be combated with vaccines, but then there are antivirals as well that are
right,
different from vaccines.
right. Yeah. So, and and antiviral is kind of like almost think about it as an antibiotic for a virus. Okay, it affects the virus in some ways. They can act in a variety of ways. Some of them could keep the virus from effectively getting into that cell it needs to get into to replicate. Some of it could keep it from replicating inside the cell. Uh, those would be the main things. Okay, and there's a lot of different chemistries of antivirals. They might keep it from making it copies of its own RNA effectively or its own DNA. And those are many of the commonly used ones. But they might keep it from infecting the cell well, like the influenza antivirals do. So there's lots of different types. The fact is that possibly with the exceptions of the ones we have for the herpes viruses, like acyclovir and drugs similar to that we use for treating herpes simplex or varicella zoster. A lot of these drugs, they're not a magic thing like taking penicillin for a strep throat.
Right.
So for example, Tamiflu, which is one the public hears about a lot, or the generic name is oseltamivir, that does inhibit the influenza virus and keeps it from effectively entering cells and doing things like that. But it really only has minimal effects on the course of influenza disease once a person is sick.
Mhm.
So, um, but let's put it another way is that, okay, another one people are familiar with is Paxlovid,
Mhm.
for COVID.
Right.
And again, it's not when you take Paxlovid like that minute you feel better, like you might with uh, an antibiotic for a urinary tract infection, might have effects in hours. It does reduce the severity of infection. And it does, if you look at 100 people who are at high risk of complications of COVID substantially reduce the risk of hospitalization and death.
Right.
But I would take this all together to say that we actually have a huge gap in our arsenal of antivirals. A lot of people talk about how antibiotics are a problem and we need more antibiotics, and that's true because bacteria develop resistance to them. But antivirals, we have even less of an armamentarium and less of a defense. So uh, we really need effective antivirals for whatever the next pandemic may be, whether that's influenza or were it to be something like another coronavirus like SARS, or the hemorrhagic viruses like Ebola, Marburg virus. We don't have great antivirals for most of those. So this is a big area where it's high risk in drug development and very expensive. The markets are not that great absent an epidemic or pandemic. So there's kind of a disconnect between the market forces, and this is an area where public, governmental investment has been really important.
So let's go back to viruses for a second, and those viruses that do infect cells, and kind of talk about that mechanism so our listeners understand when we start talking about vaccines
Yeah.
and what the future holds. So these viruses have protein coats, and people got familiar with COVID having these spikes, these proteins spikes that attach to a cell receptor, a specific cell receptor, and through that attachment, they then cause the ability for that virus to enter the cell, inject its DNA, or in the case of COVID, RNA, and begin to take over the mechanism of the cell. And our vaccines actually help our immune system recognize those spikes
Yeah.
on the virus, those protein spikes, and develop antibodies to them and prevent attack, and then attach to those spikes and prevent them to attaching to the cells. That's basically how that works. And so now we're understanding how to predict what mutated viruses might look like, in the sense that we can maybe predict the changes in those spikes and then develop vaccines ahead of time to anticipate what the next version will be, because we know that COVID and all of these viruses mutate. Maybe you can talk a little bit about that and tell us how the viruses mutate, and then how you scientists respond by trying to develop and anticipate new vaccines to combat some of these mutations.
Yeah, no, it's a great question. And I think COVID is a good example of sort of both the strengths and the weaknesses of how we're able to respond to that. Nobody recognized to begin with just how quickly and effectively the SARS coronavirus 2 or COVID virus would be able to mutate and essentially change its spike proteins so that the antibodies people make in response to an infection, a natural infection, or in response to a vaccine would no longer work as well. So, as we all saw, there was a lot of enthusiasm. These vaccines, they're highly effective. This is it. It's going to be game over. And then very quickly, these variants started developing. So even though those vaccines were made very quickly and based on the exact sequence of the original COVID virus circulating in humans in 2019 and early 2020, very quickly the efficacy went down as these new variants emerged. So in response to that, again, a strength was, well, they could quickly make a new RNA and a new vaccine to match what's circulating. But that's been like whack-a-mole, so first there's
Right.
one and then the other. Now, fortunately, there's also cross-reaction between these. And so whether from natural infection or from vaccine, the population gradually had less severe effects to each new variant, and
Sure.
immunity, more general immunity against the whole family, built up in the population. And that's largely why we don't now, when we see new variants, have the death rates that we had before. I mean,
Right.
1 million deaths. Um, but what this has pointed to, and we also knew this from influenza, which has a whole different way of varying. It essentially has 8 different segments of genes in it. They're each individual pieces, almost like human chromosomes. And those can reassort. You can get one from one influenza virus in another human or animal, and it can reassort with what you have. It's like they're recombining their genes all the time. It's like you took a big ball of ping pong balls and you could pick any of anything, and it's very easy to change. So we already knew from influenza that we're not very good at predicting what's the next virus going to be. And we saw that with COVID too, which was very different, but the same kind of challenge. And so a lot of attention as we built up things like our ability to understand the structure of proteins, what regions of proteins that your immune system recognizes and makes antibodies to, and how viruses mutate and evolve. The idea has been to try to better design vaccines to deal with either lots of different mutations that could arise. So could you find protein structures, whether they're real or even artificial ones, that could generate immunity more broadly? So
Right.
maybe you could find a protein that would get most COVID variants, or an influenza hemagglutinin, which is the main thing that our host response to is to an influenza, that could get all these different hemagglutinin types. So a lot of effort has gone into sort of computational science to say, could we either design or identify parts of what is a virus or looks like a virus that could stimulate an immune response that would sort of variant proof a vaccine.
Well, before you go on, I want our listeners to understand what roles mutations in DNA and RNA
Yeah,
play in producing these variants. So, just so
yeah,
listeners understand, when RNA gets into a viral RNA gets into a cell, it produces more of these spike proteins that end up, enveloping the new um, viral particles. But during that process, that RNA can mutate and produce a different protein or a variation on that original protein. So it's not exactly the same protein that was used to enter the cell in the first place. And it's, small mutations within the genetic material of the virus that mutates and causes these variants to be produced. Is that correct?
yeah, absolutely. So, um, you know, backing it up a little bit, I think I mentioned before that a key to the virus, its genetic information, is either DNA or an RNA. And there's RNA viruses and DNA viruses. Influenza and COVID are both RNA viruses where their genetic information, uh, their genes are essentially RNA. Some other viruses are DNA, like the herpes viruses. Now, a really important thing is that the material for copying DNA and replicating it in the cell is very accurate generally. And mutations occur, but they're at a relatively low frequency. Okay, so that's why in humans, our genomes are fairly stable among our families, and we certainly have genetic diseases, but it's unusual for new serious ones to arise constantly. I mean, part of it is our reproductive rate and all those things. But the copying material for DNA is inherently pretty accurate and faithful. For RNA, which is copied in this essentially in the factory of the cell using these proteins called polymerases that put the little pieces of the little letters of the RNA together in that chain that spells the genetic code. For RNA viruses, particularly, those are often error prone. Okay. So for routine use, it doesn't have to be perfect for the virus. In fact, it may be advantageous for the virus to generate mutations. Because when you think about it, let's say you have that spike protein, and routinely, every time your virus is reproducing in a cell, it's producing variants in the gene for that spike protein. Okay. Now, if you go into a population or a human that has antibody against one type, but maybe you produce 1 in 100,000 viruses that are very different in that part that binds to the antibody or binds to the cell. You could do what we call escape the immune system by your change. So it's kind of like, let's say your immune system, let's say, recognizes a virus by its code. Let's say we're thinking of a person, and every time we see them, they're wearing a particular coat or they look a particular way. We're looking for that person. And if that person's going to harm us, we're going to get out of their way or take defensive action. However, if that person's got a new coat or a new protein that binds to our antibodies less effectively, we're not going to recognize it. And so it's advantageous for this RNA replication in these viruses to be somewhat error prone. You don't want it so error prone that the virus isn't going to replicate. But throwing out all kinds of variants can protect you against the immune system and against the broader immunity in the population as a whole. So it's essentially a defense strategy.
On the part of the virus.
On the part of the virus that causes us as humans a lot of problems.
Right.
So that's the idea to find things that would, so this doesn't happen, for example, with measles virus, where generally it doesn't have tremendous variation. And one vaccine, that's now from, 50, 60 years ago is still efficient for virtually all measles. But so as getting back to it, there's a lot of effort to say, could we make vaccines that would be broadly reactive and protect against these kind of variants that we anticipate? And I'd say that all these things like computational biology to predict protein structure, a lot of very amazing microscopes and ways of doing structural biochemistry to say not just what is the RNA sequence and then the protein sequence of this antigen, the antigen being the thing that our immune system sees, but what does it actually look like to the immune cell?
What does its physical structure look like?
It's complicated structure, like what particular molecules are binding to what part of what receptor on the cell surface, and can we engineer those so that antibodies we make will react against all these variants. And I would say there's progress, but people have been working on these issues for a long time, albeit without artificial intelligence and immense data that we have now.
Right.
But I would still say that Mother Nature can be damn tricky. And people started talking about universal influenza vaccines more than 30 years ago, and we've still had very little progress. So it's the right thing to aim at scientifically, but I think it may be naive to think that we're going to accomplish what the human immune system couldn't and find something that recognizes everything and is effective at killing it. But that said, I think we will be able to probably improve the breadth of what vaccines protect against. But it may not be everything.
Right. And a good starting place is, I'm glad you mentioned that, is having a healthy immune system in the first place, because I think we even learned with COVID that many people in the population who had a healthy immune system were able to combat COVID through our immune system. And the vaccines certainly were an important tool in that arsenal. But human beings having healthy immune systems is extremely important in, of course, combating any disease.
Yeah, I mean, absolutely. So I think the flip side of that is increasingly because of medical advances, people are living longer lives, but they have diabetes that may affect your immune cell function. They have cancer and get chemotherapy. They have immune diseases, so they get drugs that suppress their immune systems. So, a substantial proportion of the population, or the, once people are typically in their 70s and 80s, which of course people are living longer, luckily in doing that, often with a high quality of life, but their immune system is also getting older and it doesn't respond as well to either vaccines or the infections themselves. So that's a whole field. How do we, for people whose immune system can't respond normally, how do we improve that? There are tricks about how you make that. This is another reason we need antivirals, by the way, but there's tricks about how you make vaccines that can stimulate the immune response. There's things that called adjuvants that may stimulate it more, including in the elderly. But that problem is far from solved. But I would want to add one thing that I think is very important for listeners to think about, which is, it is true that the immune system is very important, but it's also true that even healthy young people die of influenza, died of COVID in the first wave. So when people don't have immunity and they get these serious infections, you can still die. And of course, that's going on with unvaccinated people with measles now. Um,
Hmm.
you know,
Yes.
uh, Most of the children that die of measles in developed countries like the United States, where malnutrition is not as common as in many places of the world, we still have our problems with that. But most children dying of measles, if you go back before there was vaccine in the United States, for example, in the 1950s, when hundreds of people a year died of measles and there were hundreds of thousands of cases, most people hospitalized and dying of measles were healthy and their immune system was healthy. They're just were the one in 1,000, who had a
Mhm.
severe outcome and died.
Right. Understand. And that is important to understand. So this is kind of a social psychological question. You still have a lot of people even in the context that you're talking about, who are anti-vaccines. And even though there are a tremendous amount of YouTube videos and scientists and people that are explaining how vaccines work and what they are, that still persist in thinking vaccines are unsafe or that they cause the very disease that they're supposed to be combating. Now, you worked in public health. How do you approach communicating to people the safety and effectiveness of vaccines? Is it just a matter of explaining how they work, what they are, what they do to the human body, or is it something deeper than that in terms of the world views that people just hold on so tightly to?
Yeah, it's a really important question. And, when in thinking about, for example, a future influenza pandemic like H5, which I've been trying to help get the world more motivated and prepared around, or any pandemic, it's really important to recognize that we could have a great vaccine, but if people don't trust it, or if we don't have a public health and medical care system that can deliver it, it doesn't matter. So science in the absence of trust is not going to go anywhere. So I think that there are several issues. I think, you know, one, that's we're really living in an exceptional time where vaccines themselves and public health have become politicized and sort of part of political belief systems that people hold as a whole.
Mhm.
Um, and so that makes this much more entrenched than the normal distrust that people might have of certain things, industry, uh, medicine, period,
Sure.
et cetera. Um, and I think that makes our task particularly difficult. But and you have the fact that people no longer just turn to authoritative, uh, validated sources of information.
Right.
Um, they, we have social media and YouTube, et cetera, that basically reinforce whatever people saw and liked so that we all, it doesn't matter what part of this political spectrum we're on, we all risk seeing just certain kinds of information and not seeing alternative views. So I think in the long run, that piece of it to solve about vaccines and trust in public health becomes a deeper question we have to solve for democracy as a whole, which is how do we have ourselves be open to information and for people who aren't, which could include all of us, you
Right.
know, how do we then address that in our outreach? And I'm struggling with that. However, what I can say is I believe that if you respectfully answer people's questions and don't just say, I'm a scientist, trust me, I'm a doctor, trust me, um, I'm a government, trust me, but more try to understand what are the questions people have and reach them through trusted messengers, whether it's people in their community, uh, religious leaders, healthcare practitioners. Um, it is much more effective in getting messages across. And I can say with my patients, so I went through, I mean, I throughout my whole career, and I still spend a lot of time taking care of patients and seeing patients. And I would say on vaccines, including COVID vaccine in the last several years, but also other vaccines, I take a very open-minded approach where I say to a patient, for example, are you interested in getting X vaccine? It might be recommended for you at this time. And I let them tell me what they're thinking. And then if they say yes or no, I say, do you have questions about it? Or if you're not interested in it, explain to me the reasons or let me answer any questions you might have,
Right.
because that's your decision. But, I want to share with you, answers to those questions so you can make the best decision. And people will ask interesting questions. They may perceive that their risk of the disease or something happening is low. They may perceive that the risks of the vaccine are high. Um, but at least we can engage in a discussion. And I would say and then I don't say, well, you need to take this vaccine or blah, blah, blah. I'll just say, why don't you think about these things? And if you have more questions, ask me. Or if you decide you want to get it, you can talk to the nurse on the way out, you know. But
Well, I tell you that that is clearly the best approach in what we attempt to do on Nature and Science podcast. I even try to, for example, avoid using the term calling people antiviral or antiviralist because it has a pejorative connotation. And I think we have to be careful in our language and we have to be respectful of people's points of view and we have to learn how to listen. And that's exactly what you're saying. And that's so important. I'm so happy
and,
that you're saying that.
and I think in a way we need to scale that up on a social level.
Mhm.
I do think ideally you'd have CDC and state health departments working with local people and practitioners and giving them the tools, whether it's videos that dispel myths or that answer these questions, et cetera.
Mhm.
But we have to confront that, right now we may not have a public health system and spokesmen who do that. So it becomes more important. And, I'm grappling with this both in
Yeah,
what I do.
I understand. Well, with the time we have left, I know you want to get the word out about H5N1, and again, so our listeners understand the H5N1 refers to the protein structure coat around the virus. That's the way you professionals designate the different viruses or the variations. So that's why we hear
Yeah.
terms like H5N1. And prior to that, there was H1N1,
Right.
for example. So talk a little bit about what the avian virus is and at the same time, how these viruses jump from animals to human beings.
Yeah, sure. Can I get one more point in on the last?
Oh, absolutely. Please.
Because I don't want to forget it. But I think one of the hardest things for scientists to do and explain clearly, and for government officials, is what we don't know. There's this idea, and also there's a little bit of, I, I think it gets to talking down to people sometimes.
Hmm.
There's this idea that, oh, that's too complicated.
Right.
But I always feel like it's really important to say what we don't know, you know, and that doesn't, you can still make your recommendation. But for example, at the beginning of COVID, when COVID vaccines were new, we had to say we don't know yet how they will affect transmission, or we don't know yet. We can see they saved lives over these several months of these studies, but we don't know, how long that immunity will last for. So sort of, and this same kind of issues about the outbreak. People didn't know what kind of mask you need or et cetera. So I think moving, besides being personal and answering people's questions that they really have, being very candid about what we don't know, or we don't know if there's only been studies in 20,000 people. And that's a lot of people. That's a lot of studies. But
Right.
we don't know if there's some rare adverse event that's going to occur in one in
Right.
100,000. So that's also something we have to introduce more clearly in scientific communication. And I'll just leave that alone. Now,
No, I appreciate that. Thank you.
influenza. Yeah, so as you said, so most of the world's pandemics, or many of them, at least in recent history, have been influenza pandemics. And we had the great pandemic in 1917, 18, that was millions and millions of deaths in the US and globally. And what's so different there is, even unlike COVID, as I mentioned, an influenza virus can pick up a completely new piece of RNA from another species, like pigs, birds, etc., and mix that in with a human virus. And it can change its characteristics. And it can change it into something where this H protein, the hemagglutinin, which is what is H5 in running around mostly in birds now, but also cattle. So we can pick up something like that from an animal virus. It can combine into a human virus and get characteristics that allow it to grow well and cause disease in humans. And if we haven't seen that, if most of us in our 10 to 70 years of life haven't previously been exposed to something that looks like that H5, it's suddenly like a brand new virus, like a COVID. So it's not like influenza anymore. It's like, oh my God, what is this? Okay. And our immune system doesn't have cells that remember how to fight it. So we can have a wave of disease that can be very severe because there's no good cross-reacting immunity, whether it's antibody or we also have T cells that help kill virus infected cells. So that's called a reassortment. when we get a gene for hemagglutinin moves from one virus to another, and when this human virus acquires a gene associated with animal influenza that we don't have immune memory to. And that has often led to pandemics. And they can be fairly mild, like the 2009 one, which because we had some cross-reacting memory, or fairly severe, like the 1917, 18.
So
So,
to make this clear
yeah,
to our listeners, so there could be a farmer who's working with his cattle who gets infected with the virus from the cow, which isn't infectious to him, but he, as a human being, may have a virus in his body, a type of flu virus, for example, in his body, and the two viruses can interact with each other by recombining their DNA or combining their DNA and produce another type of viral particle that is infectious to a human being that farmer could then pass on to his wife, who could then go to her quilting club and pass on to her friends in the quilting club. Is that sort of how it happens, or tell us how that happens?
yes. So I'll give the H5 as an example, but right now, that's a virus, H5 influenza, that's been very efficient at infecting birds, wild birds, poultry, etc.
Mhm.
It binds, we talked about before, like the spike protein of COVID. The equivalent in influenza is this hemagglutinin protein, the H5
Mhm.
itself. It binds well to the cells of birds, okay,
Mhm.
but not well to the lower respiratory tract or lungs of humans.
Of humans,
Okay.
understand.
So it's kind of interesting. So what we see in poultry workers, et cetera, is they don't generally get like respiratory infection and pneumonia. They've gotten conjunctivitis or irritation of their eyes from presumably direct contact of their eyes. Um, and that's also happened with people in general with the cattle, because that virus is with an H5, that hemagglutinin is not very good at binding to our respiratory tract receptors. So two things could happen that could change that. Okay. And one is what we talked about before, like with COVID or mutation. The more this virus is replicating in cows and birds, et cetera, that are around people, the more chance there is for some mutation where just some letter in that genetic code changes. And we know there are single letter changes that could change its ability to transmit and go from one human or infect a human effectively. And so there could be that kind of change. Or there could be a change where it just the whole virus recombines with a human virus. So essentially, the more this H5 bird flu virus is reproducing itself around people, the more chance there is for it to either throw out a virus that is able to infect people efficiently, or to recombine with a virus that a human happens to be infected with. And in either case, we could end up with a virus that's more transmissible among humans and that is more able to cause not just eye disease, but serious disease in the respiratory tract or pneumonia. So that's why people are worried about this. And this is the history of influenza, that many influenza viruses have reassorted with viruses in animals and have resulted in pandemics. A little bit against that happening with the H5 virus is that these viruses have been around for a long, long time, and it hasn't happened. And that maybe to change the receptor enough to infect humans efficiently would result in a virus with which we might recognize based on other human viruses. So we can't say right now that there will ever be an H5 pandemic. We can almost certainly say there will be another influenza pandemic. And H5 right now is the broadest circulating virus that we don't have much immunity to. So we should be concerned about it. And other strange things about this H5 outbreak is previously H5 influenza type viruses have been largely just circulating in birds, and humans have become infected, sometimes with severe illness, but there's only been limited transmission from one human to another. And we have not seen other species infected. So this widespread infection of cows, and then the ability of so the virus has adapted to cows, which it's never done before. And also it's infected a number of other mammals. So cats on farms have been killed, and foxes and a whole mice have been infected. And also, we always are so human-centered, but it's devastated wild mammal populations, particularly marine mammals. And nobody completely understands this, but seals, et cetera, have had mass die-offs from this H5, and we haven't seen that from other H5s. So there are ways that we don't totally understand where this H5 is adapted to at least some mammals. So combining that with if it's circulating in proximity to people in agricultural settings, has got most experts at least saying we need to be better prepared. We can, this is something our society does not do well. We either get totally hysterical and say the sky is falling, or we do nothing. This is a case where we should be doing something in between. We should be getting ready in case we might need a vaccine. We should we should use it as an excuse to develop antivirals. We should use it as an excuse to test our public health system. We should use it as an excuse to rebuild trust in vaccines. Not that we need an excuse. We've got measles telling us that. But we can do all that. And if this pandemic doesn't come, that's great. We'll be better prepared for whatever the next one is, whether it's H5, another influenza, or even something else, because the science and public health practice that we really need to build, and which right now we're tearing down, unfortunately, but which we really need to build. If we do that with H5 in mind, it will be an insurance policy for other things as well.
Well, I totally agree with what you're saying. And I mentioned to you earlier, I think, in one of my correspondences with you, that I know Dr. Ward and his colleagues at Scripps Research Institute are working on what you referred to earlier as understanding the structures, the physical structure of some of these spikes and proteins, and in this case, the H that's associated with this virus. And they are trying to anticipate what this is going to look like. And so I guess my final question in this regard is, how, on a scale of 1 to 10, which may not be fair, but on a scale of 1 to 10, how prepared are we? I mean, have we learned our lessons from COVID and from other pandemics? How prepared are we?
I would give us a 5 heading toward 4. I would say we were at 5 heading toward 6. What's good? We have much faster vaccine technologies. What's good? We have states that have recently responded to a pandemic. And in theory, we have public that sort of understands some of these public health mitigations more. But what's bad? People are tired of it and can't deal with it. And it's almost like this national, and then we talked about the politicization, but it's even more it's not just political, it's emotional. People can't cope with COVID, and they lump this all together. And, you know, like I saw that as part of declaring COVID is over, just in the last couple of days, the new leadership of NIH has said, well, we're going to cut all these grants dealing with coronaviruses. Well, some of them were to do exactly what you said, which is make vaccines that would help protect us from the next pandemic, not this one, but how do we design better vaccines? Some of them were antivirals that might protect us, against an Ebola outbreak or something like that. So, and then also, right now, our fastest way to develop vaccines is with the RNA vaccines.
Right.
And we need to do research to make those better, more effective, to understand them better. And at the same time, also, there's been a lot of public distrust of those vaccines. So the community needs to share and present the data to answer people's questions about those vaccines, because otherwise, if we have to turn to them for another pandemic, whether it's H5 or something else, we will have those questions out there and that gap in trust. So, for all of how unsuccessful it was and how much our economy and culture was devastated by the COVID response, there were parts that were very successful. The vaccine saved 1 million lives here and millions of lives globally. Can we do even better? Should we? Yes.
Yes.
Were there side effects of the vaccines? Yes, but many more people died of COVID than any rare side effects. So somehow we have to step back from some of the emotion and politics around here. When I first went into government, one of the things I really loved about it is public health was something that was an area of bipartisan support. There may have been disagreements about how to do it, how much to fund it, et cetera. But, I could go down to Congress and talk to people, and everybody was interested in preparing for the next pandemic. And I think that makes sense, no matter what you believe in. And we sort of have a big job of explanation and rebuilding trust. And also, we have this big opportunity. What's all the great science, like you mentioned, computational science around how you design vaccines and antigens. Amazing scientific opportunities. The idea that we could make a new vaccine in less than 100 days. The idea that maybe we could use computational science to design new antivirals. This is the time to at least keep our investments in the future of science. And I would say it's also true about the future of public health. How do we understand what people's questions are and be sure that we don't disregard them, but answer them?
Absolutely. Well, Dr. Goodman, thank you. I've taken up a lot of your time, and I know you're still a busy man working on behalf of the public and getting the information out about H5N1 and the possibilities. I think some of the things that we talked about here today will further educate the public and hopefully help them understand. And they can certainly go to our website at Nature and Science Podcast, where we have actually information on viruses and how people can get a better understanding of these types of things and make more informed decisions. I want to thank you very much for taking the time to join me on Nature and Science Podcast today.
Well, thanks so much for inviting me, and it's really been great talking, and I appreciate your effort to share information and help people stay informed.
Thank you for joining me on this episode of Nature and Science Podcast, where science does care what you believe. In our next episode, we will hear all about chemistry and the marriage between the arts like dance and poetry and science. Chemistry is everywhere, especially in the foods we eat, So join us as we make chemistry fun with Dr. Zapra Lehrman. If you enjoyed this podcast, please hit the like button. You can become a member of the podcast at www.patreon.com/Nature and Science Podcast. Be one of the first 10 members to sign up to receive a Nature and Science Podcast coffee mug. As always, stay safe, share happiness, and remember, never stop questioning. Curiosity has its own reason for existing.